All right, this is Unit 3, the first part of Unit 3, Part 1. Unit 3 is on the fundamentals of the nervous system and nerve tissue. Later in Unit 3, I'll also talk about the autonomic nervous system. Now, nerve tissue is what controls and regulates a large number of the functions that are occurring in the body. The whole idea here is that you have sensory input constantly being monitored. You have different stimuli that can be picked up by sensory nerve tissue. And the stimuli can come from outside of the body or inside the body. An example of something that comes from outside of the body would be light, images, or sounds, or taste, or pressure, or heat. All of those can be picked up. Stimuli that are occurring inside the body that are monitored are the positioning of your body parts, the degree of stretching or relaxation of your muscles, blood pressure, blood pH, things like that. All of that information, all that sensory input is being collected and sent into the central nervous system. So you have interpretation of that sensory input. So there's an integration. You have sensory input and then you have integration of that information. And then if necessary, you will have motor output, impulses that go out to effector organs and activate those organs if appropriate. So what you see here in this diagram is this idea of sensory input. Actually, there's probably two types of sensory input going on here. You have not only the sight, the image of the glass, but you also have the sensory input occurring of thirst. So there's two types at least sensory input, one from outside of the body, the image of the water, and another from inside the body, which is the sensation of thirst. And that sensory input is picked up by sensory nerve tissue, and the information is sent into the integration center, which is the brain and the spinal cord of the central nervous system, and if appropriate, motor output. And that motor output would be a neuron, a nerve cell that stimulates muscle to reach out and take the glass and drink the water. Okay, so that's a very simple example of that kind of sensory input, integration of the information, and motor output. You do this all the time, and you do it in thousands of different ways, and you can do it instantly almost, or very quickly, like in this example, or it could be a long-term thing where the sensory input is not a glass of water, but coarse material that you're studying, and you're sending that into the central nervous system, to the brain, and you're deciding and interpreting, and then you go to an exam and you have motor output. And in that case, the motor output would not be here, like drinking, but holding a pen and writing the exam. So in terms of organization of the nervous system, the entire nervous system, there are two large anatomical parts. One is the central nervous system, which is made up of the brain and the spinal cord. And these act as an integration center, as I've said, or a center that can send out commands. And then there's the peripheral nervous system that carries information either into the central nervous system or out of the central nervous system. What we have are paired sets of nerves. Nerves are bundles of these extensions coming from nerve cells, and they can either originate at the spine or in the brain. Those are the cranial nerves in the brain, or the spinal nerves coming off of the spine, carrying messages to and from the spinal cord and the brain. Sensory information going in motor output going out. So we had two anatomical parts, the central nervous system and the peripheral nervous system. And if we just look at the peripheral nervous system, it has two divisions, two functional divisions, sensory division and motor division. The sensory information is afferent, that means going towards the central nervous system. And motor division is efferent, it's coming out. Impulses coming out of the central nervous system. The sensory division, that picks up sensory information, incoming afferent information has two parts. There's sensory afferent fibers and visceral afferent fibers. These sensory afferent fibers carry impulses from tissue like the skin, skeletal muscle, and joints to the brain. Okay. And the visceral afferent fibers, they transmit impulses from the visceral organs to the brain. So we're talking about organs within the thoracic cavity and the abdominal pelvic cavity, but also other organs, sensory organs that pick up, that are non-skin, muscle, or joint, that are picking up information. So sensory afferent fibers and visceral afferent fibers are two separate parts of the sensory division. And the motor division, and I'll talk more about how it breaks down in a minute in the next slide, but it transmits impulses away from out of the central nervous system to effector organs. That motor division has two main parts, the somatic nervous system and the autonomic nervous system. And I've broken down the way the nervous system is organized in these, sort of splitting in two and describing it in this way because it helps simplify it, I hope. It's a complicated system, and I hope this can help simplify it in your mind. So in terms of outgoing signals, that's the responsibility of the motor division. And there are two types of outgoing signals. One is formed by the somatic nervous system, which controls skeletal muscles. Skeletal muscles are voluntary tissue. You decide whether or not to contract your muscle or relax your skeletal muscles. So this is a somatic nervous system. It's a consciously controlled system. Okay? The autonomic nervous system, the other type of motor division, the other part of the motor division, is involved in subconscious control of three effector tissues. Okay, somatic nervous system here was in conscious control of one type of tissue, skeletal muscle tissue, and the subconscious autonomic nervous system, subconscious control of smooth muscle, cardiac muscle, and glands. So you decide, you get stimulus coming in to your central nervous system, you process it, and you decide that that some action needs to be taken. Your central nervous system decides some action needs to be taken, then it'll have an efferent outgoing signal through the motor division. And if it's a signal that's conscious, you're aware of and conscious and it'll be going towards skeletal muscle, so that's the somatic nervous system. If it's subconscious, you're not actually aware of it. It's happening, but you're not overtly aware in any way. Then it's a autonomic nervous system output of signal going to smooth muscle, cardiac muscle, or glands. All right, those three types of involuntarily, subconsciously controlled tissues. And just to, I guess, make it a little bit more complex, but I'm trying to keep this simple. This autonomic nervous system, which we will talk about towards the end of Unit 3, has two divisions: the sympathetic division and the parasympathetic division. Okay. Now, let's talk about some of the cells that you can find in the nervous system. So this is histology of nerve tissue. You know, there's not just nerve cells, neurons are nerve cells, but there's also many supporting cells. So there are two principal types of cells in the nervous system. The neurons, which are excitable cells that can transmit impulses, electrical signals, impulses, and there are supporting cells. Cells that surround these neurons and wrap themselves around them and help feed them, support them, guide them, many things, supporting cells. And there are quite a few different types of supporting cells, and I'll talk to you about some of them. And I'll give you some idea of the fact that neurons can have different structures depending upon their role in the body. All right, so in terms of supporting cells are called neuroglia in general, or glial cells. They have two names, so either one's good, neuroglia. They are found in the central nervous system or in the peripheral nervous system. Remember, the central nervous system is made up of the brain and the spinal cord. Peripheral nervous system are the nerves that go in and out of the central nervous system, either the brain or the spinal cord. Supporting cells or neuroglia provide a supportive, physical supporting scaffolding for neurons. They can separate and insulate neurons. They can guide neurons to proper connection as they're growing, and they promote the health and maintenance and growth of the neurons. Some of the types that are found include astrocytes. Astrocytes are found in the central nervous system. They're the most abundant, and they're highly branched cells. They cling to neurons. What you see here in the diagram is an astrocyte. There's the main cell body of the astrocyte. There's the nucleus of the astrocyte in the middle. And they have many projections coming out that can reach out and wrap around, and you see it in this diagram. It's wrapping around these capillaries, blood vessels, and also wrapping around these extensions that are coming from a nerve cell or neuron. You see here a neuron right there. There's the nucleus of the neuron in the middle of the cell body, and there are many long extensions going out. They only show one here, but there are many long extensions going out from that neuron, and they connect to other neurons or other types of tissue. And the astrocyte, which is here, I'll just label the nucleus of the astrocyte. The astrocyte that is there, pictured there, also wraps around these extensions of the neuron. Essentially, they're supporting and bracing. They're anchoring the neurons to the nutrient supply coming from the capillaries, the blood vessels. They can help guide new cells into the appropriate place, etc. So these are important cells found in the central nervous system, the spinal cord, or the brain. Here's a larger picture. I should have used this. There's the astrocyte. There's the nucleus of the neuron, and the astrocyte has extensions reaching around and wrapping around this capillary and also around this capillary. And I'll do it in a different color, reaching out and wrapping around this extension coming from the neuron. Okay, those were astrocytes. You also have, besides astrocytes in the central nervous system, in terms of supportive cells, neuroglial cells, you have astrocytes. You also have microglia, and the microglia, the microglia and also epidermal cells. Microglia are small cells with many processes. They are phagocytic cells. You would hopefully remember that there are phagocytic cells in the bloodstream called monocytes. When they go into the tissues, they're macrophages. These microglia are macrophage type cells found in the central nervous system. Macrophages of course, are phagocytic cells. They're able to engulf and take in particles like bacteria or dead tissue, and they're involved in cleaning things up. They'll help to monitor and maintain the health of neurons. Epidermal cells, also found in the central nervous system, have a characteristic shape. They're lining the spaces of the brain, and there are spaces in your brain and your spinal cord that are filled with a fluid called cerebral spinal fluid, and the epidermal cells are the cells that are involved in production of that fluid. So we have macrophage-like microglial cells that are involved in protecting phagocytosis, maintaining the health of neurons. And here you see one microglial cell embedded between some neurons, and ependymal cells that line the cavities and produce the cerebral spinal fluid, or CSF. Okay. Now, another type of cell that is supportive, and it's found in the CNS. And so this is the fourth one we're talking about. We talk about astrocytes, microglial cells, and ependymal cells. The fourth type are oligodendrocytes. Now, oligodendrocytes are highly branched cells that wrap around fibers coming from neurons in the central nervous system. So they're formed by that wrap, that insulating wrap around where they wrap themselves around the nerves or nerve fibers. They're forming a myelin sheath. That's what's called a myelin sheath. So here you see some neuron extensions, fibers coming from neurons, and this oligodendrocyte here, and I'll just highlight the nucleus. That oligodendrocyte, there's this cell body of it right in the middle, but it has extensions, and those extensions are wrapping around and insulating nerve fiber extensions. So part of its cytoplasm is actually, going out and then wrapping around this nerve fiber extension, and this one here is wrapped around there and there and there, and that cylinder that it forms wrapping around those extensions of neurons or nerve fibers, those are called that's called the myelin sheath. Now it's in the central nervous system. In the peripheral nervous system, the neuroglia, remember neuroglia are the supporting cells we talked about. In the central nervous system, we've talked about astrocytes, microglia, ependymal cells, and oligodendrocytes. Now, in the peripheral nervous system, those are nerves going into and out of the brain, and those are the cranial nerves, and the spinal cord, the spinal nerves. You have neuroglia called Schwann cells, and you also have neuroglia called satellite cells. And the Schwann cells, they also wrap around nerve fibers, but in the peripheral nervous system. And the satellite cell will wrap around just the cell body, not the extensions of the neurons, not the fibers that project out from the neurons, but around the central cell body where the nucleus is. So Schwann cells are wrapped around the extensions, and the satellite cells are wrapped around the actual cell body. So here you see in the center, and I'll highlight it in green here, there's the nucleus of a neuron. Here's an and it extends out and goes this way, and also goes along this way here, and branches like that. So that's a neuron in the middle there. And these are its extensions that I'm just erasing now. And around that neuron cell body are satellite cells, wrapped multiple satellite cells wrapped around. So it forms a covering like that around the nerve cell body. And the other type, those are satellite cells, and the other type of cell is Schwann cells, which form this myelin sheath, although it's different from the oligodendrocyte sheath in that each Schwann cell, there's a single unique Schwann cell right there, all wrapped around and there and there. Each of these bumps on the fiber or bead-like structures is a separate Schwann cell, together forming a what's called a myelin sheath. So those are Schwann cells and satellite cells. All right, let's get on to nerve cells or neurons, which are the main functional and structural units of the nervous system. Now, essentially, the nerve cell is made up of a cell body with a nucleus in it, and a single large extension from that called an axon, and usually many smaller extensions outwards called dendrites. These cells are very long-lived. Most of them live the entire lifetime of a person. They are amitotic, which means they don't undergo mitosis. They don't divide. So they are typically amitotic. Once they've developed, they don't divide further. And they have a very high metabolic rate. They have a high turnover of nutrients and energy production because they use a lot of energy. The plasma membrane of a neuron is quite unique in that the plasma membrane is on a excitable cell, and it can function in a electrical type of signaling. So you can get impulses generated, and this allows for cell-to-cell signaling. Here you see an example of a neuron. This happens to be probably a motor neuron, a neuron that will end up stimulating muscle. Here is the cell body. All of this is the cell body with the nucleus in the center. And this larger extension has quite a large base to it here. This larger extension is called an axon, and that part of the cell is involved in sending signals out away from the nerve cell. These other extensions, all of these smaller ones, all of these, they're called dendrites, and they're involved in collecting signals or receiving signals from other cells or other tissues. So what we have is a cell that can receive incoming signals via the dendrites, or sometimes actually on the surface of the cell body it can receive signals, and if it gets stimulated enough, it will send an impulse or a signal out along its axon all the way to the end. And this axon, for purposes of the slide, is fairly short, but it can be at this scale of a cell like this size. This axon could potentially be 100 feet long on this slide versus a cell body of about an inch across. So I'm just trying to make it proportional and scale it up to what this size is. So these axons can be extremely long. As we look at the cell, you see the nucleus, the cell body, also sometimes called the soma. The dendrites, you see the dendrites are indicated as being receptive regions, just as the cell body is a receptive region. This base of the axon is called the axon hillock. And here is the axon extending outwards all the way to the end to the axon terminals. All along the axon, the length of that axon, you have many individual Schwann cells that are wrapped around. The space in between each Schwann cell is called a node of Ranvier. So at each of these points, there is a node of Ranvier. Okay. The cell body contains the nucleus and nucleolus. There's a lot of biosynthesis of molecules going on from there. It's the center point from which the processes, the axon and the dendrites grow from. It is not undergoing mitosis, so it is amitotic. It has a well-developed rough endoplasmic reticulum. There's lots of protein synthesis going on, and the rough endoplasmic reticulum is called, in this kind of cell, is called nasal bodies. And at the base of the axon, you have an axon hillock, a rather large, if this is the cell body, the axon hillock is quite large and forms the base of the axon that extends out. Those processes or extensions from the cell body together form tracts in the central nervous system and nerves in the peripheral nervous system. So you get many axons or extensions going out, and they are bundled together. You can have hundreds of them, and they're bundled together into nerves in the peripheral nervous system. If it's a bundle of axons in the central nervous system, it's called a tract. So tracts in the central nervous system and nerves in the peripheral nervous system. The extensions, there are two types of extensions, the axons, as I told you, and each neuron will have one axon, and multiple dendrites. The dendrites are fairly short, highly branched processes. They are the receptive field for input from, signals coming from other neurons or signals maybe coming from other types of tissue. And the signals are conveyed as inward towards the dendrites or the cell body, are conveyed as what are called graded potentials, not action potentials. Action potentials are only in the axon. So, and we'll talk about this later, what a graded potential is versus an action potential. So impulses move through a nerve cell and can go to another cell or another type of excitable cell like a muscle. Their incoming ones are graded potentials, and outgoing will be action potentials. The axon structure is a slender process of uniform diameter, except for at the very base at the axon hillock. They're very long nerve fibers, which can get bundled together into, if there's many of them, there's bundled together into a nerve in the peripheral nervous system. Usually for each neuron or nerve cell, you have one unbranched axon, which ends in axon terminals. That's branched at the end of the axon, and that's the secretory portion of the neuron. At the end, you have the axon terminals. Also, they go by other names. I'll get into that later. But that's where a neurotransmitter will get released if an impulse goes down the axon. The function of the axon is to generate and transmit what are called action potentials. And when they do, and the action potential travels all the way down to the end of the axon, then they will secrete, as I said, neurotransmitter from the terminals. There is movement continuously of components of cells along the whole length of the axon in both directions, down towards the end, towards the axon terminal, happens to be called anterograde, or back towards away from the terminals, back towards the cell body, which is called retrograde movement of particles, but never action potentials. Action potentials only go in one direction, down towards the axon terminal. If you have a neuron with an axon like that, I'm not going to draw the many dendrites, but if it gets cut, then all this area here will disappear, all that. So it'll quickly decay. The rest will be all right, but the extension from the point where it was cut will decay. So you can imagine if you have a whole bunch of axons bundled into a nerve, dozens or hundreds of axons in a nerve in the peripheral nervous system, and it gets cut by accident or during surgery, then all of that will downstream part, all of that antegrade part will decay. And it takes a very, very long time, if ever, for it to regrow once it's destroyed, for it to come back. I mean, nerves can come back, but typically they don't. Okay, so they would start growing outwards, but very, very slowly. Now, the sheath, the myelin sheath, which surrounds these axons, and as I told you in the peripheral nervous system, it's made of Schwann cells, many Schwann cells along the length. It's a very lipid-like fatty material. It's not found on the dendrites, it's only on the axon. It protects the axon and it insulates it electrically. Essentially, it prevents any ions from going in and out of that part of the membrane. It completely insulates it. Ions can only move in and out of the membrane at the nodes of Ranvier. And for that reason, and I'll explain it more later, it dramatically increases the speed of a nerve signal or a nerve impulse transmitting down the line, down the axon towards the axon terminals. It'll very much increase the nerve impulse transmission, the myelination with Schwann cells. So, as I said, in the peripheral nervous system, you have Schwann cells, and the Schwann cell wraps around the axon and encloses the axon completely at that one spot where it's present, and it will have many concentric layers of membrane as it wraps around and around and around. The last portion on the outside is called the neurolema, and that's the part where the nucleus is. So in this diagram, you see an individual Schwann cell here, and there's another one, and another one, and another one, and each one represents a Schwann cell that during development that attached to the axon at that point and wrapped itself around repeatedly. So what you see here is the kind of wrapping that you get during the development of a Schwann cell. It attaches and starts to extend around the axon. So it's constantly wrapping itself around and around. Maybe I should have done it this way, starting here. So it's wrapping around and around like that. And finally, you get many concentric layers of the myelin sheath, and the outer portion has the nucleus, and actually that's right, indicated right there. That's the nucleus of the Schwann cell, and that outer portion is called is termed the neurolema. Okay, as I told you before, there are gaps between adjacent Schwann cells. Those gaps are called nodes of Ranvier. Nodes of Ranvier. I wouldn't use this term here. It's rarely heard or used. Gaps in the myelin sheath between each individual Schwann cell. Now it's possible if you have a axon like that, and Schwann cells along here, and you have nodes of Ranvier in between, it is possible to get what are called nerve collaterals at times, growing out of these areas, but that's fairly rare, but that's where they would come out of. There are also unmyelinated axons. Schwann cells surround the nerve fiber, the axon, but don't coil around, and those are considered to be unmyelinated, because you didn't get that, sorry, if this is the cross section of the axon here, and the Schwann cell is wrapping around, you know, normally if there's a myelin sheath, it's multiple layers of the Schwann cell wrapping around, with the outer one being the neurolema. But if it doesn't wrap around, if it doesn't coil around it and just forms a single layer, then that's considered an unmyelinated axon. And with that type of Schwann cell, you can enclose, a single Schwann cell could enclose multiple axons, but it's not considered to have a myelin sheath. These are unmyelinated axons. Now, for the axons in the central nervous system, they can have a myelin sheath formed by oligodendrocytes, which we mentioned before. Remember, the oligodendrocytes are only found in the central nervous system, and they are able to wrap around multiple axons, adjacent axons, and there would be nodes of rhombi, but they'd be very widely spaced from where one oligodendrocyte is versus the next, can be quite a large area. And there's no neurolema, because the axon itself is the cell body itself is separate from the axons. There's the cell body, and it's forming extensions out towards axons and wrapping around these axons here and here. So the neurolema, of course, in a Schwann cell would be the cell body wrapped around at the particular site. These oligodendrocyte processes or extensions reach out to coil around many axons, up to 60 axons. Okay, in the central nervous system itself, overall, if in the central nervous system, in the brain and the spinal cord, you see areas that are called white matter. They're more whitish in color, and other areas that are called gray matter. And the white matter, what it represents are collections of many fibers or axons that are myelinated. So these are, remember, in the central nervous system you have tracts. In the peripheral nervous system you have nerves. A whole bundle of axons in the peripheral nervous system is called a nerve, and a whole bunch of, or bundles of axons in the central nervous system are called tracts. So the white matter represents these tracts. They are dense collections of myelinated fibers, axons. The gray matter is mostly soma, or cell bodies with nuclei, and a lot of and some unmyelinated fibers. The white matter is myelinated fibers, and the gray matter is cell bodies mostly with nuclei. Okay, now I did mention we looked at a drawing of one type of neuron, which is probably a motor, depicts a motor neuron, but there are many types of neurons, nerve cells in the nervous system, and they can be classified based on either structure or function. When we look at the structural classification system, what you have are three types: multipolar, bipolar, and unipolar neurons in terms of their structure. Multipolar have 3 or more processes, extensions, quite abundant. Bipolar have 2 processes, an axon and a dendrite, and these are fairly rare. And unipolar have a single short process, and it can be divided into 2 areas, peripheral and distal. I'll talk about these later probably. So we have these 3 types: multipolar, bipolar, and unipolar for structural classification of neurons. In terms of functional classification, we have sensory neurons, motor neurons, and interneurons. The sensory neurons are the ones I talked about for sensory input. They're in charge of sensory input into the central nervous system from stimuli either outside of the body or inside the body, but the impulses move towards the central nervous system. This is the information that your nervous system is continuously collecting about your external and internal environments. The motor or efferent outgoing type of neurons sends signals out away from the central nervous system. Remember, the central nervous system is structured that you have incoming sensory input, afferent incoming sensory input, then you get processing in the central nervous system, and then, if appropriate, you have motor or efferent output. Interneurons will shuttle signals inside through the central nervous system, different pathways within the central nervous system. So they're more involved in once those sensory signals come in, then we're talking about shuttling those signals through the different parts of the central nervous system with interneurons. So for structural, we had multi, bi, and polar and unipolar, and then we had for functional classification, sensory, motor, and interneurons. And here you see a table showing, a multipolar type of cell with the cell body and its nucleus and many dendrites carrying incoming signals to the neuron, and then a myelinated axon that sends impulses out. Remember, action potentials are sent down from the neuron, down the axon to the axon terminals at the end here, where you get secretion of neurotransmitter. And you see in blue there the Schwann cells forming the myelin sheath. A bipolar cell is shown here, where the cell body is in the middle, and you have an axon, a dendrite on one side and an axon on the other. And then you have a unipolar type of structure of neurons, some neurons, where you have a receptive area, and the signal goes, the whole thing is considered a axon, and the signals are going in this direction down towards the axon terminal. This is the receptive area receiving signals, and the cell body's in the middle there. So these are some examples of cells that have the multipolar or bipolar or unipolar characteristics, and I'm not going to get into the details of this slide, and there's more shown here, but these kind of details we'll get into later in this section in later parts of this unit. So that's the end of Unit 3, Part 1.